Our next speaker is Seth Harris. He is a distinguished scientist and director of computational structural biology and drug discovery at Genentech. He's been there for most of his career, but before that, got his PhD at UC Berkeley and a postdoc at UCSF.
Thank you. It's a real pleasure to be here, and I think it's worth spending the first minute just saying I have a bit of a soft spot for CCP4. I remember as a grad student new to crystallography, enough time ago that I don't like to think about it, but quite a while ago, being introduced to CCP4 as a kind of magic toolbox of things that seemed really amazing. And not only the tools themselves, but the chat boards where quite a bit of the personality of the people involved seemed to come through, whether it was lighthearted and amused teasing from Jared Klavecht to other people, or concise but somewhat exasperated answers from Paul Emsley about, have you actually read the manual I provided? Or I think there was a quote from one Eleanor Dodson that I'm paraphrasing a bit at the time, but saying something like, oh, multi-crystal form averaging knocks mad phasing into a cocked hat, which had enough Britishisms in it. It took me a while to figure out what was said. But the point of this being that it's not just for doing science, but also the learning of it. And kind of over the decades since, it's great to see the longevity of CCP4 and the project and the community and all those personalities and characters behind it. So with that, as I said, my path has landed me at Genentech in the structural biology group. And I've moved from kind of experimental crystallography, which I still do, and now EM with our terrific EM group, into a computational side. And I lead a small group within structural biology that's focused on the computational angles. And in this talk, I wanted to share kind of two major parts. I was asked to talk about macrocycles in drug discovery, but I also want to introduce and build to that with a description of the platform we've built to kind of analyze structures and basically help anywhere structural data is used, not just in generating it, but interpreting and analyzing it as well. And so this kind of SOUL platform was our name to give it a central system, particularly around this GYST part, a guide for structural thinking, guide in Mars that I'll touch on a bit. So those two major sections, and I'll finish with a few comments about kind of where we're headed with some of that downstream. There's many motivations why I kind of got steered towards or was interested in computational aspects. This is pre-AlphaFold, but just feeling that a lot of things were changing in the compute power. But one of them was maybe also just I wouldn't say laziness, but I like being efficient. And in industrial structural biology, you're often told like, oh, great, we did a KRAS structure. How about 400 more? And your eyes kind of glaze over. Like, how many times am I going to build the same thing? And so that scale of data we had too was shifting from, instead of just being lucky to have a structure protein, we now had hundreds. And when you're new onto a project and inheriting something like that, how do you navigate hundreds of things and digest or distill that information, the gist of it, as it were, into something that's manageable and easily interpretable? And the other part was that, the chemists look at a structure and they almost ignore most of what you built and look at just these interactions, which is important. And then the structure, I'm torn between saying it's carefully filed away or it's thrown on the pile roughly, but we end up with this big pile of data. And as these computational tools were coming along, it seemed good to be able to extract information from that mountain of prior structures, both the PDB and our in-house ones, in a way that could kind of provision these things into machine learning and AI. And so this GYST system was meant to be something of kind of a Sherpa for your exploration of these landscapes, using this kind of analogy to mountaineering or something, where you're working your way through these landscapes of structural information, all those different sizes and shapes that you know about, but providing kind of some of the heavy lifting for you, especially in routine areas that we're doing repeatedly by hand otherwise. So this is a kind of a gist of GYST, as it were, but essentially we were absorbing all of the in-house structures we had, all the PDB, and with the advent of AlphaFold, some of the computational models as well that kind of earned a seat at the table once the performance was good, and parsing all those into kind of what are their parts, which chains are which are ligands, looping through that and establishing relationships between those, and providing kind of pre-built computation, which meant that if you wanted to know about conservation, ligand interactions, or anything else, it was done for you already. You didn't have to do that by hand every time you pulled a structure off the shelf or out of your database. And with those relationships, the idea was that it could eventually provide suggestions or information, which is kind of where the point we're at now, is how good are those suggestions and things it's providing to you. But all this was meant to also be integrated between that kind of 1D, 2D data world into the three-dimensional molecular frameworks that we're pretty familiar with. These numbers are a bit out of date. There's actually now 1.1 million individual macromolecular chains in the collection between mostly in the PDB. And so that was the part I was saying, like a lot of the annotations that were done are usually done by the experts kind of by hand and put into their PowerPoint files, but they weren't often stored along with the structures and certainly not with the functional data. So the idea was, can we unify all of that together And now you can start to do, see patterns and conformational changes that correlate with the activity relationships that we're getting. And so this was meant to be something of a virtuous loop between analyzing structural information as that is fed into the compute and new algorithms and methods come out. Those can be sent back to the users who may not be experts in those areas, but could access those algorithms much more easily this way, and then get new insights, which could further build new models, back and forth in that way. And so I won't go through all the details of these. This is kind of the 4 components of our platform. We have a limb system on the left, GP2S, for Genentech protein to structure, that tracks our experimental workflows, but really talking here about GYST, the annotations of structures, and then GYDE and Mars. And the other part is that we're working on open sourcing some of the components of this, GYDE in particular at the moment, which is the part where you do engineering, design, and so on. And so it'd be great to be able to kind of share that with the community rather than just keeping all those proprietary things tucked away. And then there's some underpinnings to all this that our team has built as well. And so this is just a quick synopsis showing that it helps people in all the places structures are used, as I was saying. And so it's people who come, I don't know if I call it traditionally, but you're interested in a certain protein, you have one structure you want to go look at, you can go do that, but it's now in a very enriched data context. So in the past, it was like, here's the coordinates, good luck to you. Now it's more like, oh, here are the coordinates, here are sequence relationships, here's conservation, here's the pockets, here's the interactions, et cetera, et cetera. And those are all available kind of in single clicks to paint those onto the structure from that database. Furthermore, as you've seen, and we know, we often want to do a whole collection of structures and have them aligned together. So GYST was made to kind of easily give you the infrastructure to make custom collections of proteins or all the structures known for that protein, and provide those, which are then aligned to the same reference space and easily sent to Mo or PyMol or 3D environments. And then there's the protein level knowledge as well, which is kind of like a collection, but pre-made for you. So if you're interested in EGFR or something, this has a compendium of all the information about EGFR, the sequence analysis, et cetera. And the other important part is the whole API. So not just the kind of traditional end user who wants a GUI, but all these kind of computational people who are coming to the field with their machine learning AI, wanting programmatic access to that same data. And so you can ask these questions and say, I'd like to see all the PISA interfaces that you've tabulated across all these proteins and seek out the ones that are of a certain size or character or properties, or ligand interactions that we were feeding to machine learning colleagues who are looking at these affinity models. And we can provide that with analysis of kind of what's the quality of the electron density for each of those ligands with it as well. And that's also come about to fuel this Mars platform, which is our viewer for all that GYST data about proteins, pockets, and so on, ligands, but at a proteome scale. So it's a way that we use all that information in target assessment to decide which ones look like feasible drug targets. And this plot here is kind of the whole proteome scored in a somewhat empirical scoring system we've developed to kind of chart some of that. And I'll touch on that briefly as well. So this is, again, just trying to move quickly through it, is showing you an arbitrary structure from the PDB of this GPCR. And it's complex that I could, in the GYST page, pick each chain, send different information into it onto the structure. So one of them is colored by the conservation down here from conserv. This one is the PISA interfaces. This one grabs from uniprot disease mutations that might be relevant for disease. And lo and behold, you can see instantly that a lot of them are clustering to the interface with this protein. Maybe that's why they're pathogenic or not. And then this one's colored by surface exposure. Like all these things we annotate are very easily sent in. And again, you have them all kind of indexed across the sequence as well as the full information from that available in these different areas. And we'll talk a bit more about the ligand interaction one, because that's obviously one of the focus of this workshop as well. I would say too, it's not just the overall conservation, there's pairwise species. In the drug discovery, we're often interested in what's the people say, oh, is this the same in mouse or not? Because with this antibody binding here, we want to know the safety species, is it conserved? And so you can project those things too, species by species, onto that as well. And so this is just another version here with KRAS showing that you can kind of drape those layers of data onto the 3D world in single clicks very quickly. I like to think of it as like David Attenborough, navigating how a bumblebee sees flowers in infrared or UV, but now it's proteins with these layers. If anyone does a good David Attenborough voice, our next video, it'd be great to have that. It'll be like, the little protein didn't stand a chance. Or something, I don't know, sorry. And then this was just the quick synopsis too, to give a feel for it. But all of it said that GYST information, when we first built it, we're like, wow, if anyone wants to come along and know about all the proteins at once, And sure enough, our target qualification group said, hey, we want to know about all the proteins at once. We're like, oh, say less, here you go. So we had this API that was able to send all that raw data. And then we've also worked with that team on developing a scoring system to try and get things to, the appropriate weights in terms of what it means to say, oh, there's a good structure. There's only an AlphaFold model. What are the pockets like? What's the coverage of the structure? What's the resolution? Are there homologs? And then in the druggability side, we're pulling information from Chembl. This is pretty analogous to open targets, if you're familiar with that as well. It's an open sourced version that's doing the same type of thing. And we pull in some of that information too, these kind of TCBI categories where things that are biologically known, have chemical matter known, are in the clinic, or this T dark and red. It's a little mass, but the big clusters down here. This axis is tractability from low to high and druggability from low to high here. So, it emphasizes precedence, which means if people have worked on something, it tends to score highly. If it hasn't been worked on, it tends to score lower. It doesn't mean it's not just as good as those. So you have to kind of understand how the scoring works. But it's very interesting, I think, to navigate the genome this way and the targets in it, and to determine what you think is going to be pretty easy to work on, where the challenges may lie if you tackle something that doesn't score as well in some of these categories. All right, and so this is just kind of speeding up a bit here, showing some of the tracks on a typical page in GYST for a structure. I'm going to use USB7 quite often. It's a deubiquitinase, but it's all indexed across the sequence. You have PFAM domains, what's actually in the coordinates. The gray parts are gaps in the coordinates, conservation, as I mentioned. But down here, the piezo interaction, protein-protein interfaces. As you know, it can do that it does that for crystal symmetry as well in crystal structures. And so you can pull up all those, you can zoom in, see the sequence names and all the particular weights of how much buried surface area, in this case, is what I'm plotting. And on the left, the actual details are all available for the interfaces. This is between a macrocycle and the protein. So you can see those interactions. You can sort them by buried surface area. So if you were wanting to do like a mutation to break an interface, you could very quickly find places where you might get the best bang for your buck to design those mutations for that. Again, a lot of this is not rocket science. We're using tools that are out there. It's really just bringing it all together so you can do it in practically real time and do these things as fast as you're thinking of them or keeping up with a seminar speaker. You can almost kind of replicate their slides as they go, and then you can ask really annoying questions about some specific amino acid because you're looking at it too, but that's not really the purpose of it. And so the 3D part too, as I was saying, if you click this little, what we call the olive, it's really meant to be like a highlight patch. It's a little mushed here, but it sends that information onto the 3D structure, colored the same way it is here, and you can change the coloring, send it again if you want to change that. You can do the different interfaces, and that shows you where the interface was between those residues and the macrocycle ligand in that case. Similarly for small molecule interactions, I kind of titled the talk, All Ligands Great and Small, a little nod to that veterinarian show. That's a bit agnostic about what the animal is like. Is it a small molecule, a macrocycle, or a protein? Interactions are really the fundamental piece of biology we're trying to modulate. They, of course, have different tools that go into them. This one, we're using this track here is annotating protein small molecule contacts. And these are from a tool called View Contacts in Proasis, a third-party tool. If you zoom in, you'll see they're all kind of labeled with a type of interaction on that amino acid. In this case, there can be multiple ones per amino acid. So the sending, this is the little legend. You can change the coloring or what you see on each one. But when you send it over to PyMol, you see each of those little blocks becomes its own dashed line. So in studying this ligand, you can very quickly kind of see maybe where the better and worse interactions are, where you may want to extend it out as well. And if it's complicated there, you can see in the objects that are made in PyMol, you can turn off all the van der Waals at once if you like, or other things, and clean it up and view. So again, just kind of trying to be very streamlined and versatile in how those are viewed. And this is a quick view of making collections of aligned structures. You can do that with a sequence-based one, at which point it can become auto-updating when new structures come into the PDB. You'll get a little email saying it's been added, aligned, presented for you. But it's helpful to then start looking at conformational differences across the structures, clusters of water, so on and so forth, with the ligands. The other part when you have a collection is you can measure ad hoc things that you like in the distances or angles or dihedrals. So it's a way of making kind of a conformational landscape that we've heard a bit about. This one, you're picking what you want to measure. So in the instance of kinases, people familiar with that, there's a DFG, there's a helix C, kind of conserved features that indicate active or inactive. But with those measurements, you can make a kind of quantitative view across all the structures compared to your reference. and then see very quickly differences between, in this case, chain A and chain B of this kinase, and how the helix sees position and so on. What's interesting there is if you know there's about 8,000 kinases in the PDB, and if you were to try to read all the papers to figure out if your structure is novel or not, it takes a while. And so the point here was Henrik Mobitz at Novartis had made some nice dihedral measurements that were kind of indicative, these DFG -1 +1s of the conformations. And so in one go, your new structure comes in, gets measured, you can land on this kind of landscape of all 8,000 kinases and tell you whether you have a fairly novel conformation or not on these particular metrics. And of course, there's other ways to do this without choosing what you measure, variable autoencoders and things for structure conformations, but they're a little less easy to interpret kind of why it's distinguishing things. All right, I'm going to kind of move ahead. I don't know if this timer's going, which way it's going, up, down. And just show you, in terms of the interactions, the other thing we've done is on a mass of structures, in this case HSP90, it can say across all the structures, here's an aggregate of all the ligand interactions that have been observed. And so this track here, sorry, it's a little small, but this little heat map kind of says every ligand interaction, we're taking account of how often it touches that residue. You can see a couple of residues in particular light up. And if you paint that track on the structure, you get a very detailed view of the pocket where ligands have touched it. Again, you have to kind of filter out a bit. There's a lot of ligands that are maybe not as interesting to you, but you can ultimately trim those down and see kind of an interaction map on the structure that way. Of course, as soon as you see that, you're wondering, well, which structures are doing what? And so we have a broken out version of that aggregate where this track now has every individual structure in a row. And you can see on these little tick marks where that one has a ligand touching those residues. And MOE has a function like this in the PLIF plots, as they call it, protein ligand interaction fingerprints. But we did this also for pocket predictions as well in some other areas. But it's nice then to see if you're studying a ligand that has a kind of fingerprint or pattern like this, you're trying to think about where you need to extend it. All these light colored ones mean that there is a ligand somewhere, not this one, that does touch that residue. So you can scan down and find other ligands that are going to areas that your ligand doesn't and bring those in very quickly. So again, it's a tool to navigate large structural cohorts of information, hone in on what might be interesting for you in drug discovery or designing better binders in those fronts. This one too is just showing that if I switch from individual structures to that similar HSP90 case, but here for USB7, it's an aggregate view of information. And in this case, it's a track that has the public structures that are available, and they're all lined up to the sequence as well. So you can see kind of the domain boundaries and the extent of those structures. Those can be colored by a bunch of metadata, so the resolution, the space groups, and so on. Just showing some of that here to organize and find structures. So if you have a new one that's in a new space group, it's pretty quick to find relevant fragments in those same regions. This is colored by method. There's an NMR in orange, some EM ones in purple, and the rest are crystal structures. These EM ones too, you can see they look like full-length structures. As we know, some of the EM things, the model itself may cover a much smaller piece. So we made another version of this track that's actually the atomic sequence, meaning what's in the coordinates, not just what's in the secret header. And some of those EM ones end up being just the C-terminal part as well. So it's a bit more true to what's really in the structure. And I thought it was a bit silly to color by title, for instance, which is available in the metadata, but it does help find kind of groups of structures that come from the same study or show you things that have other proteins bound as partners in those complexes. So I just want to then touch on this guide piece. So after you've kind of done studying your protein of interest, if you actually want to do something with it in terms of engineering and design, you can ship collections over to GYDE and look at the sequence selectivity around the pocket. But it's also where we set up a place to run AlphaFold, Boltz, Chi, your thermal MPNNs, protein MPNNs, many of these emerging tools that are out there. I was built first with some of the antibody tools as well. And so just to kind of briefly touch on that, because I think it's a really fast and changing area. But there's a lot of folks, I think, that wouldn't be comfortable running it themselves. And so this is one of the ways to do it. But the real benefit, I think, is not just running the tool. There's many ways that you can, the interfaces people have built to run those. But how do you actually navigate the results again? It's that question where these tools tend to spit out huge amounts of information and data. You could have 1,000 new protein designs, for instance. And so these things have built-in plotting, 3D viewers, and sequence and ways to import other functional data. So you can very quickly kind of lasso things on a plot and pick which quadrant of information you want to store. So this is a part that we're about to kind of open source as well. And this is just showing some of the menu, again, already a little out of date. We have bolts 2.2.1. This is AlphaFold 2 because we're in industry, so we can't use 3. But you can kind of put in very quickly any of the new tools coming out that you like, and then use the infrastructure of the data viewers that's in there to do that. I would just kind of really quickly show a toy example. So the Baker Lab published an engineering effort on TEV protease, which is kind of a dog of a protease, want to improve its efficiency. So they tried keeping, they were basically going to run protein-MPNN to redesign it, keeping conserved residues the same and only allowing to vary things away from the substrate site that were less conserved, either 30, 50, or 70% sets. I showed you in GYST this conserve track, and you can kind of artificially color it with a cutoff around 5. And if I just in one click paint that on a structure, you can see it kind of replicates here. So if you're doing this on a new protein where someone hasn't nicely provided for you their selection in their supplementary information, you can kind of do this in seconds and build up a similar one. This part too, near the substrate peptide I showed you, we have the PISA tracks, so you can very quickly also select those out, leave them out of the selection, and then go over and run protein-MPNN. In our interface, we've added a few bells and whistles of what you can tune in that. It runs really quickly though, and I think as you just heard about too, for some of those methods, you end up with kind of getting your newly designed spaces, and we showed that kind of within this run, you can in a few minutes kind of replicate what was done with that with TEV. And the other part, again, like I'm saying, if you get this result, you're like, okay, great, I've got hundreds of things. How do I pick what I want? We have the sequence logos here to show you kind of positions that have the most conserved recommendations for redesign, and also a frequency widget with a slider. And so you can lower this down and get kind of the population of what was recommended at each site, or if you just want the most strong recommendations, you can filter those down that way, and also hand pick what you want and send those off then for the next steps in the construct synthesis and usage. So that was kind of that first part, but really, I think what it just helps with is having all that pre-computed data at your fingertips means you do things that you might otherwise have been like, it's going to take a while, and you just have it there. It opens the door for some serendipity and testing things and noticing things that you don't otherwise do. I think also as we head towards some of the new machine learning AI groups that have joined us as well, they're like, oh, we're going to do it all. We have this chat interface. I'm like, I'd really love for you to keep some of the viewing of data that's in that 3D context, right? I don't want a paragraph of a chat bot telling me, here's 50 sites, and you're like, I don't know where they are. I haven't memorized the numbers. So we're trying to make sure that there's some kind of hybridization between these methods and keeping some of the really powerful things that our users like and enjoy in the visualization we've done so far. And again, we're happy to work on this and try to open source it as well. So that I want to segue into the macrocycle discovery part as a special class of ligands. And I think you'll hear more about that in the next talk too, on the bicyclic versions. But they're basically like a cyclic peptide, as people probably are pretty well aware. I think every talk I've seen about this always has a slide like this. I borrowed this from Shannon, a postdoc in my group who works in this area. But they're essentially in between bona fide small molecules that go up to about 500 to 700 Daltons, and large molecules, which would be antibody and antibody-like things, which could have just one domain around 50 KD, but the full antibody is 145. And so they benefit from being, antibodies typically are injected and only are going to access things outside the cell and control, cell signaling targets, your EGFRs, like Herceptin here. Or on the small molecule side, you have oral bioavailability, and they're taken typically as pills, but you don't have as much screening capability. DEL is a good way to access pretty large screening here. And so these have the benefits of having strong screening capabilities, whether it's phage display, mRNA display, techniques like that. But still also being kind of potentially intracellular, although permeability is a big challenge in macrocycles. And then kind of the diverse chemistry of the small molecule space opens the door to a really huge, essentially infinite space, like 10 to the 60th types of combinations of things. These are just 3 quick examples of ones that are kind of in the process of commercialization. Luna 18 from Chugai, just showing like kind of after you discover them too, the amount of diversity in linkages, tags, whether it's for solubility or trying to get into the membrane, or bridges and ways to cyclize those as well. It's pretty a wide space. I won't go into too much more on that front. A lot of the stuff we've been doing has been kind of like what PeptiDream established. So this Sugo lab had developed these flexozymes, which are engineered tRNAs that allow you to redesign the genetic code and incorporate non-canonical amino acids. So now you can imagine this kind of in vitro translation system with these, where you can kind of, instead of putting in a tyrosine, put in something else, and a huge variety of possibilities there. When those are done and made, you essentially have your mRNA library you've constructed. The in vitro translation turns that into this peptide with those non-canonicals, but it remains attached. You then do your binding screen, and all these things that do bind, for instance, the yellow and the magenta, have their little tags, everything else is washed away. And so you amplify it out, and the tag tells you kind of which thing was the binder. You do a few rounds of that and see the enrichment you get for that. So that's kind of one of the methods of macrocycle discovery for those first hits. And so one of the places we've published on that last year in conjunction with Aaron Duber's lab at Genentech was looking at USP7. And the theme of this was a bit that there are some small molecules inhibitors for this deubiquitinase, but it took kind of years to find them and years to optimize them. And Aaron was showing that in kind of one cycle of macrocycle discovery, you could get nanomolar inhibitors from this that could serve in their own right as a therapeutic, but potentially also would they serve as kind of a guide to light up for you and canvas the protein surface and find where small molecule development might be efficient and effective. And so we were comparing somewhat the kind of space and style of the macrocycle binding to small molecule binders, as well as the response of the protein on those fronts. And this is one of the places where macrocycles in GYST as well, our system, and in some of the other systems, they're tricky. They're full of these non-canonicals. Our GYST system sees the amino acid name as being a non-canonical, decides it's a small molecule ligand. And we're like, no, that was attached to the macrocycle. So we need to add some more logic to get that. A lot of those small molecule interaction tools kind of choke once the molecule gets that big, and so they don't finish and tell us all the interactions they see. Or they tell you a number, but they dropped a digit because it didn't expect to have that many heavy atoms in it. So there's some problems like that, which made it a bit, as I said, a labor of love to kind of annotate all the interactions, which Shannon was doing for our help on this paper. This was just showing some of the immunogenesis at each site, and we were trying to determine whether, if you see lots of interactions, maybe that site's pretty optimized, and you wouldn't see much red here. The red meaning other sequences could get enriched if you mutated those in the macrocycle. And these other ones that accommodate a lot of changes didn't have as many interactions. We're just kind of correlating some of that biology function with the structural themes there as well. Again, that's published. I don't need to jump into too much of that. But there are certainly confounding effects. This glycine doesn't tolerate changes. It's not really interacting much, but it's a glycine. So probably it's helping with the conformational variability in the macrocycle, right? More so than specific interactions as well. And the other part, just to come back to our platform, where I was mentioning the measurements you can make, we wanted to see that the protein would respond to the macrocycles. Was it doing it similarly to the way that the small molecules do? And so this was Shannon was doing, was measuring these various features of the site. She could do that across the small molecule bound in purple, macrocycles in orange stars, compared to a known ubiquitin bound active form and the inactive. And then the orange is the alpha fold floating around. So you can see it does actually, the overall structure looks similar, but you can see the active site's doing something a bit weird compared to the known experimentals. And again, you may want to measure multiple things to kind of determine that. So I'm just going to zip forward a bit. There's kind of two ways with macrocycles that one of the big problems is handling them in structure refinement. They're complicated molecules, and making a CIF file is difficult. I don't like to complain because we have this very creative peptide therapeutics guy who's also a chemist, and he's actually made the molecule and figured out how to make it. So I don't want to complain making the digital version of it in a CIF file, but there are certainly ways that it feels right to call it a polymer and then have these building blocks that are non-canonicals. But it gets very error prone when this guy is super creative, as I said. He's made multiple rings and they're all modified. And so I tend to go towards this, it's a big small molecule version. Just give it the smiles, it makes a big CIF. Some of the tools have not, they kind of lose the information that the peptide bond should be planar and things like that. And so it's been a bit tricky kind of sorting through it again. And, you know, there's Corinna, there's Elbow, Grade, East Drug, and different ones do different things exactly. And again, that we have these problems where some of the tools, not so much the CIF generation, but afterwards, don't like these extra digits when you get like a chlorine that's now called 104 rather than there's so many heavy atoms. Anyway, all those things are just a part of it. Since we're at a meeting about the tooling workshops, things that need some attention still, I think, in our hands. I'd be happy to learn how people are handling those elsewhere. We're also doing more EM structure for this Chimera and Isolde, which I've only started kind of going into the last few years. But it has a real-time force field. It's density aware. So it's a great tool. It has its own parameterization for its force field. And for a while, the only thing that I could get to work was have in this Smiles to CIF was Global Phasing's grade tool, the older one, which took a while, but it would work. And so, these things all have their kind of a little sensitivity, but it's nice to find one that works well and then kind of explore what we can do to kind of improve that more broadly. All right, I'm running low on time. I just want to say in the closing, it's been obviously a super exciting time for a lot of these things. This is a plot this summer, and keeping up with it is obviously quite difficult. Already there's new things since then that we need to add to. And I think what's coming for us is a lot of the kind of agentic interfaces. And so we've built many of the tools that are accessing all that GYST information I showed, but now through agents. To summarize this, I'm a little conflicted when you're doing something like structured determination and you know what you want to do. And it feels like sometimes the decision-making of these chat things are like a toddler. And I don't know if we want to put in the hands of a toddler the decision, but it's a toddler that seems to be very good at accessing huge amounts of information as well. So obviously these things will keep improving. But I think as we've heard in the panel discussion in other ways, there's a lot of things coming that way. So big thanks to the groups here, especially Kieran that runs kind of the engineering and software groups we've had, and Jack, who's a really terrific powerhouse between experimental and machine learning, and some of our colleagues in these groups. And thank you all for the attention as well for that.
Questions for Seth? All right, I'll start with one. So amazing platform, very jealous of both the visualization and being able to use all these tools. But I'm just curious on, chatting with some of your colleagues, if there are tools that really seeing the interface between some of these tools have been a lot more valuable than others. Where do you see sort of synergy between tools, and where do you see ones that, are providing synergistic information?
So you mean like which tools kind of augment information in the other tools?
Yeah.
That's tricky. I don't know if I have a good answer for that. I mean, some of them feel like they're kind of their own exploratory thing and they haven't yet fell in well. Other parts like PISA seem pretty fundamental to a lot of it. And so that information works really well with kind of the cross-referencing you may have to conservation, disease mutations, and so on. Yeah, and then the kind of workflows, I mean, we bridge between structured determination paths that we're working on, as well as like the binder design stuff. And so seeing those kind of emerge, Bindcraft, which we heard about, and RF diffusion and things, I think those work nicely, that they blend to protein MPNN and the whole pathways you can build that way. That kind of speaks to it, I guess. Thanks.
Again, thanks, Seth. Some really nice tools and interfaces there. I'm wondering, in terms of the user base, how much of how many med chemists or biologists would be using this in terms of different ways to design their own experiments?
Yeah, we've kind of charted that a bit. I was happily surprised to see a pretty broad usage from many of the departments in Genentech and also at our colleagues in Basel, that were not just the structural biologists and comp chemists, which were our main power users. I'm not quite sure exactly what everyone's doing in it. We don't chart all their motions and movements, but there's certainly some appreciation from the biology side too, where they've become aware of AlphaFold and other things, and they want to kind of dive into some of those details. So
yeah, and I guess the follow-up question is, if you've got people, non-core users, sort of more from other areas, what are their needs, or what have they requested additional features that you guys haven't thought about?
Yeah, we certainly get that too, and we just ran out of development time to do everything that everyone wanted, so we tried to prioritize. But there's things around construct design that came up a bit, and quite a bit on the biology side. The target tractability and discovery is also a big area that's a lot of people are now kind of focusing on. But we haven't finished everything we want to do, so it's kind of jumping around. Thanks.
Great platform. I'm jealous as well. But one of the great things about CCP4 is that we've got people at various stages of their career. So imagine I was much earlier in my career. Do you have any thoughts about, with the access to all of these tools, what do you feel people should think about when they're doing their research? Because there's so much you could do. What should people think about doing?
Yeah, I mean, I can't answer that specifically on each tool. There's probably different recommendations, but it's certainly a philosophy that comes up where we've met people that say they want to democratize access. And as soon as you do, they say, no, 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 not to that, or not for those people. They're not smart enough, or, I don't know what they're thinking. But there's definitely this weird kind of tension between how much job security a comp chemist wants and how much they want to share, or I don't know what it is. But it feels like they're also worried that putting things that can be quite sophisticated to the hands of people that aren't in that field could lead to global apocalypse, or, I don't know what the worry is, but there's certainly ways that you could maybe go down the wrong path for quite a while if you're not familiar with what it's recommending. So we're trying to put in enough documentation and enough office hours and expertise or examples to suggest to people ways we've thought of using it, but also leave it open for people to try and explore. And hopefully it's not damaging. I mean, we're not, holding nuclear codes here. We're trying to design new things. So that's been my philosophy, is to make it as open as possible, but with the caveat, like you're saying, that it can be a bit overwhelming, and we're trying to provide enough recommendation and thought behind it to give some amount of steering or guidance. But
So you're telling people to read the documentation?
I don't do that, but they probably should.
All right, let's thank Seth again.
Thank you.